Okay, so welcome everybody. It gives me a great pleasure to welcome our seminar speaker today, Professor Camille Maumet. She has a PhD from Inserm in France, a postdoc in the University of Warwick in Coventry in England, and then also at Oxford in the Nuffield Big Data Center. And she's currently a group leader, again, at Inria in Rennes, I understand. She has been a driving force behind neuroimaging data modeling, and specifically topics around reproducible data processing and analysis and imaging. She's been a pioneer in neuroimaging analytical flexibility and has very, very active roles within the Organization for Human Brain Mapping and INCF, the International Neuroinformatics Coding Facility. So it is our great pleasure to welcome her today as our speaker. Please go ahead and share your screen.
Thank you very much for the introduction. So I'm going to share my screen.
I should say that if you have a burning question, feel free to put that to speak up and mention it during the talk. Otherwise, most questions we'll keep till the end. But if it's something that is important during the presentation, I have the chat open and I can keep an eye on that.
Thank you very much for that. Well, hi everyone. It's my great pleasure to be here today. And thank you, Celia, for the introduction. I'm going to talk about reproducible neuroimaging, and in particular about reproducibility when we look at data that were processed with different analytical pipelines. And in the following, in my introduction, I will give more background as this to focus. But first, my research in general, is based, it's based on a phenomenon that started about 10 years ago that has been sometimes named the reproducibility crisis. And basically 10 years ago, researchers and scientific communities realized that it could be more difficult than we would have thought to reproduce findings that were published. And I've included here a few examples of reproduction rates that come from biomedical sciences. And of course, these low reproduction rates were seen as an issue for multiple reasons. Not only because first we might ask if it was, and I'm including myself obviously, if it's a good use of our time if we produce research that cannot be reproduced. The same holds for the money that is put into research. In biomedical science, the reproducibility crisis has often been seen also as an impediment to translation into clinical practice, which is often not a short-term but a long-term goal of our research. And also this phenomenon might be one of the drivers of reduced trust in science, both in terms of the general public, but of research communities. And of course, this sounds very depressing, but I like to see the reproducibility crisis, and I'm not the only one here, fortunately, as a wake-up call for research communities to rethink the way we do science. And in fact, that reproducibility crisis has led to best practices in terms of how we do science, and in particular, it has led to a new field of research that we can call reproducible research. In what I'll present in the following, I'm always gonna focus on what happens in the data analysis phase. So I'm taking the assumptions that I have a pile of data. So I typically work with neuroimaging. That's why I have brains as small examples for data sets here, but obviously this would apply to any type of data. And I typically work with MRI, magnetic resonance imaging. So I have a pile of data that come from an imaging instrument, an MRI in my case. I'm going to do an analysis on this data, and this is going to give me a result that I'm going to publish and share with the scientific community. So in the following, I'm going to represent that pipeline with this smaller schematic. So here, the first round is the data, the arrow is the analysis, and then we get our final results. And so why should we care about reproducible research? I do really like this field because it's really interdisciplinary. When I talk with people from many different fields, when I talk about reproducibility, this is something that is meaningful to them. But what I found really funny is that depending on who I'm talking with, from which community, they might have a very different view of what we mean actually by reproducibility. So going back to our examples where we're doing data analysis. So for instance, when I think about people who care about how the brain works, such as neuroimagers, when we do publish a study, such as the one represented here, what we really mean when we publish these findings is that we hope that they will remain true in a different context. So using a different data set and a different analysis. So for instance, let's say my study is demonstrating a new biomarker for Alzheimer's disease. When I publish that, my hope is that I'll still be able to observe this biomarker if I look at a different set of participants. If my acquisitions are done in a different hospital that probably has a different MR system, the acquisition themselves might be done in a slightly different way, and so on and so forth. And that's the assumptions behind a lot of papers. And of course, sometimes results do reproduce, and we're happy we have found the biomarkers that is generalizable and that we can see in many different contexts. The issue comes when our results do not reproduce. And then I've taken the example, the extreme example of a biomarker and direct application into the clinics, but probably more realistically, this idea of facing results that do not reproduce is something that us as a researcher, we have often faced. Basically, when we build our research, usually there is a first phase where we are building on the literature and we first reproduce existing findings. And so what happens when we try and reproduce an existing finding and we cannot get the same results? Well, then we face a cloud of questions. So when I can't reproduce the original results, typically the first question that I tend to ask myself is, well, did I understood correctly? So maybe when I did my reproduction, I didn't do the method correctly, and that's why they don't reproduce. Or maybe it's because actually the pool of participants that I was studying was too different, was not from the same population as the one that was used in the original study. Or maybe it's something more specific about the tools I've been using and so on and so forth. And we have many, many, many questions. And typically the field of reproducible research has been addressing all different types of reproducibility. So we've talked about the case where we are thinking about reproducing the same results in a completely different context. We also have the case where we're trying to get the exact same results using the same data and the same analysis. And that's typically the type of reproducibility when I'm discussing with my computer scientist colleagues. And reproducible research is looking at all of this. Sorry. And there are different names for those, even though the consensus, I think there is no consensus across fields, but often the case where we are reproducing the same results using same data, same method is referred to as computational reproducibility, and the reproduction in a different context as replicability. And of course, there are all sorts of different flavors of these. So I've drawn two axes here with same or different data, same analysis, different analysis. And we could have two more cases here where we have different data, the same analysis, or the same data and different analysis. And all of this is part of reproducible science. What is an important key message here is that depending on where we observe a lack of reproducibility in that matrix, And of course, it's not only 4 points, because you have to imagine when we talk about different data, the data could be different in many different ways. It could be a different subset of the population, a different acquisition protocol, and so on. So depending on where I'm standing in my matrix, the reason that drive a lack of reproducibility might be very different, and the solutions will be different as well. And so in the following, I'm going to first start by reflecting on the type of problem within irreproducibility that the neuroimaging community has been focusing on. So starting with different data, and then we looked into the question of what happens when different analyses lead to different results. And so on our way to fix irreproducibility in the neuroimaging community, and as I was saying, our first focus, because this is the first aspect of irreproducibility that were tackled by the community, we'll look at what happens when we look at the same analysis but in different data. The first reaction, one of the first reactions of the neuroimaging community to the realization that we had a reproducibility crisis was to discuss the sample sizes that we were using. And there were a number of papers, including this one by Button and colleagues, that basically showed that neuroscience studies in general had a low statistical power. And as it turns out, low statistical power, by definition, is going to lead to a high rate of false positives in the literature. And so, of course, this is not a problem that is easy to solve, because there is a reason why we were working with small sample sizes, because neuroimaging acquisition are very expensive. We are working with human participants. It requires really a deep expertise in order to be able to do this acquisition and so on. But at least it was an observation that soon enough was understood by the community, and that the community reacted to work against this issue. So that was one aspect. More recently, people in the neuroimaging community and beyond, so here I've taken an example from X-ray imaging, where the question was to detect lung opacities. And basically what they showed is that when you would train the model using only images from male and tested on female, you would get a higher rate of false positives. And that's what you can see as an illustration in this image here, was the opacity, the falsely detected opacity is in yellow, and in fact correspond to the breast of that person, because the space in which the algorithm was trained didn't include that part as normal variability. Of course, this is a toy example, but I really like this study because it illustrates really well the bias that can arise in our models if we are not careful enough about how we are building the samples we're going to train on. And so this is a question of the lack of generalizability of the model we are building. And this issue is even more acute if we think about the case of underrepresented groups. And that's the question of fairness. Because by definition, if we take a representative sample of the full population, there will be only a few cases of people from this subgroup. And of course, fairness is extremely important, especially in the field of medicine, because we don't want to have worse outcomes for, in terms of health for subgroup of the population. And so the way the community reacted to both of these issues was through proposing a common solution, which is data sharing. And so data sharing is helping here because first, it can help us achieve larger sample sizes, because by pulling data across different sites, it's easier to achieve larger sample sizes. But also because, again, by pulling data, we are more likely to include different types of variability in terms of population, but also in terms of material we've been using, and so on. And in the neuroimaging community, in the past 10 years, we've seen increasing data sharing, initially going through sharing the data that were associated with a single study, so tens of participants. Then we've seen efforts moving on where basically we had research-led efforts, consortium that focused on, okay, agreeing on a type of images people would be interested in, and then doing this acquisition together so as to build larger sample sizes. And there we reached around 1,000 subjects. The 1,000 functional connectome project was really a pioneer in that space. And even more recently, we've seen finally funders trying to tackle this issue of reproducibility and basically putting money into funding high quality resources that would be made available to the full research community to answer a wide range of questions. So that was the first reaction from the neuroimaging community, and we've seen that in other communities as well, to the question of reproducibility. And this is tied really quickly with how we work with statistics in the brain imaging community. When it comes to the questions of having different analysis or different pipelines, initially, there was much more skepticism. So in our reproducibility matrix here, we are now in the top right-hand corner where we look at the same data and a different analysis. And here you might, so I was saying there was skepticism as to whether actually different, so I'm using analysis pipeline and methods or analysis as synonyms here. And so the question was whether the way we build our analysis pipeline could really have an impact on our results. And so initially I represented the analysis pipeline through this schematic, but in principle, in most analysis pipeline, in fact, we can define two high-level steps. The first one is data preparation. This can include denoising, registration into a common space, correction for motion, and so on. Then we get some data, some measure we are interested in, and finally we can do a statistical analysis that will let us answer our research question. But in practice, often it's much more like this. And of course here I've given just a single line, but in practice it would be even more complex, like a tree. And so we have steps one after the other. In neuroimaging, I would say in an fMRI pipeline, which is the type of data I'm working with, we have about 10 to 15 steps. And these steps can be, as an example, segmentation, registration, up to the final statistical analysis in the end. And the thing is, when practitioners choose their pipeline to analyze their data, they in fact have to make a lot of choices. For each single step, typically, there's going to be multiple algorithms that could be used. Even if with my expertise as a neuroimager, I know the specific algorithm I want to use, typically it's going to be implemented and available in multiple software packages. Even if I always use the same software package because that's the one I really like, well, typically it's going to be available in multiple versions. Even within the same version, there are often a range of parameters that are considered as acceptable for that particular step. And finally, we might run that very same step, same tool, same version, same parameter, but in different environment. Maybe you're using an HPC cluster to run the subject level analysis, and then you do the group level analysis on your lab. And so this means that, in fact, instead of having one single pipeline that leads us from our input data to our final results, we have a family of pipeline. And as practitioners, we have to choose between many different options. I've included a few here, but obviously, if we start mix and matching all the different options, we very quickly end up with an analytical space that is extremely big. And so for quite some time in the community, people would consider that basically this was the expertise of practitioners to go from input data to the final results. and that this would not necessarily impact the results we got. And then there was this project that is called NOPS, which is a many analyst project. So many analyst projects are studies that are designed by a group and then submitted to different groups who work basically on the same research questions. And so this is an example in neuroimaging, but there have been examples before in psychology and social science, well, different fields. And so what happened here, the setup was as followed. So the team who led this work acquired one data set that was built to be of high quality in order, and they provided 9 research questions. And these were yes or no research questions. So in the field of fMRI, yes or no research questions are typically, is there an activation in that specific region of the brain when I do this task? And here it was a gambling task. And basically what they did is that, they recruited a number of teams of experts, 17 totals, and each team was tasked with analyzing the data, the same data, to answer the same yes or no research question. And what happened as an output of this study is that first, and I think this is an important result, first, each single team chose a different pipeline. So of course, there were commonalities, but there is also a lot of variability in there. And here on this graph, we're looking only at the red dots. On the x-axis, we can see the research questions, the yes or no research questions. And on the y-axis, we have the proportion of teams that replied either yes or no. And so what we see at the bottom here are agreement within the teams to answer no. And what we see at the top is agreement across teams to answer yes. And then we have, for a number of hypotheses where, so here I took a 20% threshold. Of course, we could discuss the threshold, but for a number of teams, we basically had contradictory results when looking at the results of different teams. And the most extreme was hypothesis 1, where we have about a 40-60 split between the teams. And this was basically started to raise questions in the communities because, of course, when we start with the same data set, we wouldn't expect experts to get different answers. And I think right now in the community, people are asking for ways to handle this variability and finally have practical solutions. And here I'm going to detail a little bit one piece of work that we did with Alex Baumring and Tom Nichols from Oxford University, where we looked at this variability, but instead of looking at the variability across experts, as what's done in the NAP study, here we looked at the variability induced by using different software packages. And before I get into more details, I just want to say a few words about the type of data I've been working with. So I've mentioned it briefly. It's functional MRI, more specifically task-based functional MRI. And the idea here is that basically we are gonna ask the participants to do a specific task while they are in the machine. And what we get as an output, the important here, so the background is just a picture of a standard brain. The important bits of information are the yellow to red clusters, and this corresponds to brain areas that are activated during the realization of the tasks. And of course, we can look at all sorts of different tasks. And here we have, for instance, a visual paradigm, hand motion paradigm, or listening to music. And depending on the task, we're going to have different activations that may overlap or not. And so the question that we wanted to answer here was basically to investigate the variability that we would observe in our fMRI results, so our activation maps, when using different software packages. And in order to do so, we reproduced 3 studies from the literature using 3 fMRI software, AFNI, FSL, and SPM, because those were shown back in 2012 to cover about 80% of the fMRI literature. And we worked to align the pipelines across software to make them as close as possible, given the parameters that are made available to practitioners. So we didn't modify the inside code of the software, we just used the available configurations. And what we found out of this study is what you can see here. So each of the columns here corresponds to a different study that we reproduced, DS01, DS109, DS120. On the last row, you can see the figure that we extracted from the original papers. First, you can see that these typically have different forms. They can be across the volume or on the surface. And then for each row here, we have a reproduction using a different software package. And for instance, we can see that for DS120, it was not feasible to reproduce the results of that study using FSL because the type of design that was used was just not feasible in FSL. So here I'm going to focus more specifically on the first study, DS01. The first observation we can make is that we successfully managed to reproduce the original clusters. And in particular, the FSL results are closest. And this is good news because that was the software package that was used initially. But we can see a number of differences, clusters that are more extended or smaller clusters that completely disappear or appear. And remember that here we only have one slice taken around here, but in fact, brain images are 3D images. And when we look at multi-slices, it's even more obvious that we can see differences across software packages. And so we measured that using dice coefficients that ranged from 0.3 to 0.5. Then after looking at thresholded images, our final activation maps, we decided to look at differences one step before in the pipeline. And basically by looking at unthresholded statistic maps. So these are the exact same results, except that we have values everywhere in the brain, including in the voxels that didn't pass statistical significance. And here we can see that globally, we have good agreement across software packages. Note that we could not compare with the original results because these statistic maps are not always shared. And we measured with correlations how close the results were across software packages. And so as a conclusion to this study, we had shown that basically different software could impact the results. And probably the differences are stronger the further you get into your pipeline. So we had stronger differences in activation maps than in statistic maps. And this is also results that were observed in the NAPS paper that I didn't discuss, but that you could see if you go back to the original paper. So this was the first results. And then as a follow-up work, because that's the question everyone was asking, and we were also ourselves very curious about, we wanted to better understand those differences. Because remember, we had worked on aligning the pipelines to be as close as possible across the software packages. And so the question then was, could we explain the remaining differences? And so what we did here is basically using our knowledge about the software packages, we identified 5 main factors that could have impacted, that basically were implemented differently in the software packages and could have created those differences. And so I'm going to describe this figure here. So here we go with pipeline 1 from a full AFNI pipeline. So that's equivalent, if I go back here to my AFNI result, to a complete FSL. So 4 is a complete FSL pipelines. And again, these are my FSL software results. And in between, we did mix and match using different options from different software. And here we had to write custom code so that we could plug some part of one software into the other, or change the different default parameters, which is different from what we did before, where we only focused on using the available parameterization. And here what we can see, so of course we did the different mix and matchings that were available. And here for that particular study, which is the SO1, we can see that going from here to, so we used a common preprocessing step, and then we use subject level signal in AFNI, and then we use for the rest, subject level drift and noise and group level stats, we used FSL. And we can see that going from 2 to 3, basically there is only one step that changed, which is the subject level signal, either in AFNI or in FSL. And we can see that this was an important driver in terms of changes that we can see in the activation map. And so the good news is that for each study that we looked at, basically we could identify one main contributing factor. The not so good news is that basically this factor was different for each of the 3 studies. And to me, this calls for a better understanding of what's happening in the pipeline space as to which steps are driving the differences, and maybe we should reproduce that across different data sets or across larger data sets to better understand in which context which step is going to have the stronger impact. And so this leads me to the next part, where we are trying to build solutions to better understand this, what we call analytical variability. So basically the variability we see in the results when we take different pipelines or different analysis. And studying analytical variability, I think one of the main challenge is that basically analytical variability is the consequence of very different effects. First, on one end, we have what I would call unwanted variability. So basically, when I look at 2 pipelines that are exactly similar, but were run on a different operating system, for example, and I get different results, this is giving me an idea of the stability of my tools, or the stability of the method I'm using to answering a given question using a given data. So this is really variability I would like to get rid of, or at least be aware of when I discuss my results. So that's one thing. When we talk about analytical variability, this also covers the case where we might get different results using different pipelines, but not due to the pipelines themselves, but in fact due to the fact that, for instance, we don't have enough data, or we don't have the right data to answer the question, the research question we'd like to answer. And here, basically, getting different results with different pipeline is just a reflection of the fact that our results are unreliable. In other cases, sometimes using a different pipelines, we're going to get different results just because our data violated the underlying assumptions that are made by that pipeline. So there is a pipeline to data interaction here. And then finally, the reason why we use different pipelines is often that we want to better understand what's happening in our data. So as an example, if I add age as a covariate in my statistical model and get a different result, this is going to inform me about the importance of modeling age in my data set and this impact it has on my data. And this is what I would call variability of interest, in that this is something practitioners might want to explore to better understand what's going on. And so when we talk about analytical variability, often it's a mix of all this, going all the way from like the reliability of the tools we're using, the reliability of our tools for our data, to an exploration of our data. And so in order to better understand what's going on and to tackle this issue that is difficult to explain right now, we have tried to contribute to a number of efforts in order to first describe the neuroimaging pipeline space. So in particular, in a project that is called NARPS Open Pipeline, which is a follow-up of the NARPS study, so we are trying, so initially each team in NARPS had to submit as their results yes or no answers to the research questions. They had to submit an activation and a statistic map. So those are the typical output of an fMRI study, as we've seen earlier, as well as a textual description of the methods that would follow good practices defined in the COBIDAS guidelines. And they were not asked to share the code they used to produce their final results, probably, that's my take, because this is not a practice that is widespread still, at least at that time, in the neuroimaging community, and probably we would have a reduced number of teams that would have signed up if the code would have been required. So the goal of NARPS Open Pipelines is to basically try and reproduce the code that was used by the different teams. And this is feasible because of the textual description and of the final statistic and activation maps that were shared by the users, by the teams, sorry, and can be used as a ground truth to check that we reproduce the pipeline properly. With this project, we have participated, and in particular, Boris Klené, who is the software developer behind this project, participated to a number of hackathons and had different contributors. And of course, this project is still open for contribution. So if you're into reproducing fMRI pipelines, please do get in touch. So we think that NARPS is interesting because that's one instance where we get variability in pipelines that corresponds to what experts would have used. Another approach that we've been focusing on is to try and enable provenance tracing in neuroimaging. So provenance is basically a description of the different steps that led to the creation of a data set. And so with Satra Ghosh here, we've been co-leading a BIDS extension proposal that focusing on how we can share provenance with our data sets. Just to give you a sense of why we think this would be useful, we've mentioned earlier the difficulty we can have when trying to reproduce results using what's provided in papers. Provenance can be a way to have information about how we can reproduce exactly the same results, so computational reproducibility. It's also a way to, if we want, for instance, to revisit the literature. So sometimes what is identified as a best practice in terms of parameters or tooling evolve as the community better understand the statistical approaches or the methods or the interaction with the assumptions on their data. As best practice evolve, it is useful to be able to go back into the literature and say, okay, this study was done with that parameterization that is no longer considered as a best practice. And sometimes the paper do not have this level of detail. So that's one place where provenance can help. Another use case is the case where we found a bug in a tool. And of course, neuroimaging tools, just like any other tools, will sometimes present bugs. And that's also useful to be able to go back and say, okay, this particular paper was built on this tool with that version, and this is known to be suboptimal now. So that's one other important results that we can get out of provenance. And then another one is to better understand what we've been discussing until now, the idea that different set of parameters might impact my results. And so BIDS-Prov is a draft BIDS extension proposal, as I was mentioning earlier. And of course, we are happy to get feedback on this as well. And so I'm going to finish with two different studies we've been working on to try and propose solutions for practitioners. So we've seen that analytical variability can be an issue. We've seen proposals or steps into a direction where we'll better understand the analytical space. Here we're exploring, can we deal with that variability in our day-to-day practice? So the first study that I will present only very briefly here is same data meta-analysis. So the idea here is that we have a data set. We are going to run it through different pipelines. So this is based on the assumption that someone has the expertise to say, okay, those 10 pipelines here are the one I think are relevant to answer my research questions. I get different results, and I want, instead of having 10 different answers to my problem, get statistical estimates of what's the consensus across all of these approaches. And of course, this is very close to meta-analysis, but it is different in the way that we are using the same data as input, and basically it's violating the independence assumption. And so we have some work that will be presented at OHBM this year by Jérémy Lefort-Bénard in collaboration with Tom Nichols. So that's one first research direction. Then another question is how these differences in pipeline can impact our ability to do mega-analysis in neuroimaging. So so going back to my functional MRI analysis, where I have data preparation and statistical analysis, in practice, it looks more like this, where we're going to have multiple subjects that each are going to go through data preparation, and then together they're going to combine into a statistical analysis. So we call this a multi-stage approach, where we have the subject level and the group level. Our hope is that in the future, as more and more data are shared, we might be able to retrieve directly derived data to ask new scientific questions, to build a statistical analysis to answer a new statistical scientific question. But by doing so, we are completely losing control on the pipelines that led to obtaining this derived data. And it is very likely, in fact, that subject 3 and 2 and N were prepared using a different pipeline. And so the goal of that study here was to investigate the validity of our group statistic when we combine data that were obtained with different pipelines. And this is a work that was led by Xavier Roland in co-supervision with Pierre Morel. And here, basically, we based our study on the HCP young adult data set, and we looked at 3 pipeline variations. So in very short, we looked at different spatial smoothing, different modeling of the brain response, and different way of correcting for subject motion with an increased number of parameters. And we randomly assigned subject to one pipeline or variant of that pipeline, and then we did 2 sample t-tests and counted the number of detections that we would get. And if there was no differences across pipeline, we would be completely under the null. And so that's one way for us to estimate an empirical false positive rate. And I've not included all the estimates that we had in the paper, but here is an example. So on this graph here, we can see the 0 line corresponds to a perfect estimation. Everything that is inside the gray area is within the confidence bound. What's under corresponds to a conservative statistics. So we get less sensibility, but we are still valid. And what's at the top here is invalidity. And so basically we could see invalidity when the number of motion regressors was very far away from the one of the reference pipeline, so 24 versus 0, or when using a different smoothing. And beyond that, depending on for a number basically of variants, we could see invalidity, which questions our ability to directly combine maps that were generated by different pipelines, which in my sense calls for sharing more details about how those maps were shared, so that maybe in the future we can take this into account when we build our group statistical analysis. I'm almost out of time, but I'll finish. So I'll go through really briefly through that last study. So here the idea was to leverage large and open data set. We used NeuroVolt, which is a database of fMRI statistic maps. What's great about NeuroVolt is that there is a lot of data out there, so over 500,000 images that were uploaded by different people. So it's very diverse in terms of the tasks, the pre-processing, the analysis, and so on, but only minimal metadata were required. And of course, this was one of the reasons for the success, so that's clearly not a criticism on my end. But then the goal of this project was to ask whether we can take advantage of a large pool of unlabeled data or minimally labeled data, such as the one on Neurovolt, to help answer new research questions. And so what we did, and this was work led by Elodie Germany in co-supervision with Elisa Fromont, is to build a model of fMRI statistic maps through an autoencoder that would be agnostic to the type of task, processing, and so on, and then to fine-tune a classifier to basically predict the task. And what we showed on 2 different data sets is that the model that was pre-trained with the autoencoder would always perform better, especially when the problem was more complex. So with smaller sample sizes, or when we had a number of studies that were gathered together, so more heterogeneity, basically. And so I'm going to end my talk here, and I'm really happy to answer any questions.
Thank you very much. Are there questions either from people who are online or people who are here in the room with us? If you're online, you might need to unmute yourself or put something in the chat. I don't have a visual view of everybody.
Maybe I can stop sharing and that would help.
Yep, perfect. And I'll change the view. I can start off actually by asking a question. I thought it was very interesting to your last topic, where you have the different you had the machine learning going into latent space and then coming back out. So the other thing that I thought was that it could be related there is the idea of certain pipelines or certain software packages or certain algorithms being inherently less reliable. So have you played with the Bayesian interpretation of all of this? Sort of putting a prior on something.
So I haven't worked with a Bayesian interpretation, but that's surely things we've been discussing with colleagues, trying to understand which models, and I think this applies probably more to the statistical models, or maybe to the pre-processing as well. So which models are going to explain better our data, and could we give them more weight? So that's not something I've explored myself, but certainly that's of interest. I think the closest work we're doing is the same data meta-analysis, where we're giving each pipeline the same prior, but that's that idea of like combining results together and considering that different pipelines give us different pieces of information about our research problem.
Yes, exactly. When you were working with the idea of taking the same data set, pre-processing it with several different techniques, and then creating an aggregate, that you could incorporate there the idea that some of them you like better than others, but you'd have to decide which ones you liked better, of course, which ones you would give more weight to. And I thought that would sort of be an interesting perspective, the sort of prior distributions on certain types of pre-processing pipelines.
Yeah, totally.
There is a message in the chat. There's Guda. Did you have a question, or you were just saying thank you? Whoever Guda is. Okay. The question from Darcy Crenel. I like the mega-analysis work. Is it going to explicitly address successful statistical correlations with differing pipelines on, let's say, 10 versus 1,000 participants?
So I'm not sure I understand the question. So the idea, so I'll just try and say a few words and then you correct me there, see if that's not what you were getting at. So the idea of these methods is, yes, to address the correlations between basically the input data sets, between basically the statistical maps we get from the different pipelines that were obtained using the same input data sets. But I don't quite see where you're getting at between 10 versus 1,000 of participants. I'm sorry.
Darcy, do you want to turn on your microphone?
Oh.
Yeah, I'm just asking about some results in correlating traits with um, uh, uh, few participants versus thousands of participants. You can get different results. You can get noise in the 10 participants study that you don't get in 1,000. So I'd like to see that kind of work across different pipelines. What kind of noise does different pipelines give you for that kind of study, that kind of large mega, like you said, mega analysis studies?
Okay, I understand better. So I think that's a very good point. So one thing we've been, and I think it's getting at, one thing we've been looking at is how analytical variability, like the differences we see across pipeline, varies with sample sizes. And in fact, there is published work in psychology that shows that basically as you get larger sample sizes, or it could be in terms of participant, but also in terms of like repetition, or if you have basically a stronger signal, you're going to see a smaller effect of that variability. So that's one thing I think we need to look into, because maybe tomorrow, if all our study have 10,000 participants, then this is no longer a problem anymore. I think the second point that might be related to what you're saying is the extreme case where basically that I was trying to get out with my different cases of analytical variability. Basically, sometimes we're going to see variability in the results due to the different pipelines, but that's not related to the pipelines. That's related to the fact that we have too little information in our data to answer our research problem, and basically our results are very unstable. And so that's something we're going to see if we work with very small samples.
Hi, Celia. May I ask a question?
Yes, absolutely.
Okay. My question is about the big cohorts like UK Biobank. They collected brain imaging data sets. And we know that this kind of cohorts are not primarily like collected data for brain imaging. They have like bunch of different like data regarding like different traits, for example, neuro system traits, immune system traits, and so on and so forth. So do you think this kind of like data sets which we have with like higher rates of heterogeneity have some like effect on brain imaging reproducibility results?
So I think it's, so UK Biobank is certainly one of like the data set that have been used a lot in our community because of the high sample size that was unprecedented basically when UK Biobank arrived. I think the fact that we get these large amount of data, and this is getting back to the beginning of my talk, is helping in two directions. First, because we increase sample sizes, as you said, and second, because we're going to get a better understanding of what's the true variability in the general populations, which is something we really should care about if what we want is to build models or results that will be applicable in the population at large.
Does that answer your question?
Yeah, but I'm still wondering if other traits in our dataset, for example, in the UK Biobank, affect brain imaging and or affected by brain imaging or nervous system brain, and they make different story about reproducible results when we are using brain imaging data.
So what do you mean by different stories? Different subgroup that might get different results across?
Right.
Yes. Yeah. And that's, again, I mean, getting back to what I was saying at the beginning, the question is, are we trying to represent the population as a whole? And this is a variability that we want to be able to encode, or maybe we want to define subgroup in that population and understand better how those subgroup are going to impact on our results. Surely, but I think we do need ways to like model that variability, understand it, and being aware of it more generally.
Okay, thank you.
Are there other questions from those online or in the room? I do have one more question myself. You know, there's a sort of classic phrase or classic saying in statistics, if you analyze the same data set with more than one method and get the same result, the same conclusion, that you should be far more confident in your conclusions, you know. So if the result is going to be sort of strong enough to stand out, even if you've used different methods of analysis, then it's probably more trustworthy. Does that paradigm translate into this really high dimensional space?
Sure, and I think that's something you already see in practice, like in papers. I've mentioned that people use one pipeline, but typically what they do is actually they use one and then they do variation on a number of parameters that they think might impact their results or that the reviewers ask them to do in order to check if they impacted their results. And I think, like, to me, this is a saying that completely fit with the work here. I think the multi-dimensional space is interesting for us to better understand which bits in the pipelines are the main drivers of that variability we've been observing. And once we know that, we can go back to the community and say, okay, those are the important ones that we should check when we publish our results. I'm not asking everyone to go through that huge space. Clearly, that's not feasible. But getting a better understanding of what's happening in there is, I think, where we should get at. Yeah.
I think there's a really interesting nuance in there, which is whether the sensitivity of your results to different parts of the pipeline might actually depend on the phenotype of the individual. So, you know, somebody who has Alzheimer's, maybe it's a different portion of the pipeline that is very sensitive, or somebody who has had a stroke, maybe it's a different portion of the pipeline that is very sensitive. And I don't know, is that something you have explored already?
I think so. What you're saying with phenotype, but also maybe with research question, maybe like the same people, if you're trying to, depending on what you're trying to predict, you might have one pipeline that is more sensitive or not. So I've not been exploring that. I'd be really interested into looking into whether we can first find commonalities. Are there patterns that repeat across different research questions, which we did not see in our reproduction of 3 studies, but if we work with, I don't know, larger samples, if we use the UK Biobank and different data set, can we see factors in that pipelines that repeatedly leads to high variability? Then probably that's something we should look at more closely, because then you can go back to the community, and if you are able to say that's the step you should be closely looking into, first, that's something that is doable in terms of testing, but also people have the expertise to say, okay, that's that step, you change that parameter, that's the meaning on my actual data.
Thank you. So we are at 1 o'clock. I guess there is an opportunity here for a last question or two, if anybody has one. Oh, there's one that's come in. Another question from Darcy. I like putting provenance information in bids, but I'm skeptical about what tools are going to consume that information and what they can do with that. Any thoughts about that?
So what they can do with that, I can see very clearly. So being able to, once we all share our provenance in bits, okay, that's not tomorrow, but let's say this happens in the future. The examples I was giving earlier, like we know this parameter is no longer optimal. I want to go back to the literature and see if the papers I've been basing my reasoning on to build my research have this parameter, then maybe I should have a lower confidence into them. So that's one application. But more broadly, I do believe that if we have information in a machine readable way, there will be people, including myself, trying to mine it and better understand the analytical space, which is something I think we need. So I think the tools are going to be built, because this is going to be unique information in machine readable form.
Thank you.
Yeah, I'd always meet a lot of skeptical people, so fine with.
I think with that, we will give a really warm thank you. Thank you so much, Dr. Maumet, for a really interesting talk. And I think this is going to open some lines of inquiry and thought for many who are here today. So thank you so much.
Thank you very much.